Hello my dear students, I hope all of you are very well and every day you are learning something new. So, today we will continue our learning in pharmaceutical biotechnology. This is the 30th lecture of our entire lecture series in pharmaceutical biotechnology. So, today I am going to tell you something very interesting which is related to my previous lecture. I hope you remember in my previous lecture I have promised with you all that how bacteria and smaller microorganism they share information with another. I am going to tell you. So, today I am going to that is fulfill my promise. I will teach you about the microbial genetics, how microorganisms are communicating with each other, how they are sharing the information and how a new microorganism getting a lots of advancement from their previous one. That is you are going to learn. So, what are the things I am going to tell you in my entire lecture? First, we will tell you what is microbial genetics, then I am going to tell you about the transformation, then you will learn about transduction, conjugation, plasmid and transposons. So, something very interesting thing is actually waiting for you. So, first of all, what is microbial genetics and why it is very much important to us? My dear students, you have to understand that bacteria basically these are very small microorganisms. These are actually smaller that is microbial substances, and they actually have to survive in this planet despite lots of environmental factor which is negative for them, like temperature, pressure, humidity. Temperature means del, that is why I am leaving temperature, pressure, humidity, then lots of new viruses also. Okay. So, there are lots of negative elements actually present in this planet. That is why you can see due to global warming lots of new bacteria or microbial diseases are actually coming. Basically, they are trying to change themselves in such a manner, so that they can sustain in high temperature, So, that is why they are actually sharing information with each other, so that the next bacteria get mutate or get much more advancement, so that they can resist themselves from various environmental factors. Now, what are these things or how they are actually sharing information, or if they share information, what are the things that are happening? So, here is the answer. One is the transformation, one is transduction, one is conjugation and another is the transfection. Transformation, basically the plasmid transformation. I hope all of you have listened my all the previous lectures, where I have shared with you what is plasmid. Basically, plasmids are small DNAs, they are consisting some genetic information. Bacteria are actually consisting plasmids. Those bacteria they can share the plasmid with each other, and when they share this plasmid or some part of plasmid, we call it a transformation. Transduction, basically various kinds of viruses, more if I say more precisely bacteriophages T2, they can input their genomic material to the bacteria. They can, if they can get the bacteria, they will do something, they will having some information, either resistance or something. Okay. So, this is called transduction. Another thing is called conjugation. This is happening through sex pilus. Okay, this is happening through sex pilus. And last, transfection. Here some electric shock or other things actually happening, due to that some pore is actually created, and through that the genomic material actually entering to the bacteria. These are the general overview of microbial genetics. Now, how it is playing a big role or why? So, you have to understand that the amplification of specific region of a DNA can be achieved with bacterial enzymes using polymerase chain reaction. I hope you remember in my previous lecture I have told you what is polymerase chain reaction and how we can amplify specific part of a DNA or other enzyme based on the method of nucleic acid amplification. So, if we have some bacteria and if we know that certain genomic part or certain gene of a bacteria is good for us, what we will do? We are going to use that genomic material, that particular gene or that particular segment of the DNA for our own purposes, for applied biotechnology. What do? We will try to cut we try to cut that particular area, and then we are going to insert that to the other bacteria, so that they can translate such proteins. See, for example, like this bacteria, viruses, etc., we are taking some part of its DNA, then we are actually inbuilding with another, and then we are doing some PCR or other kinds of amplifying that is activity. If I tell you more, the introduction of a foreign DNA or RNA into bacteria or eukaryotic cell, it is a very common technique in molecular biology. There are multiple ways of foreign DNA can be inserted. One is the transformation, transduction, conjugation and transfection. See, at the beginning I have told you the basics why bacteria are actually doing such kind of activities, why the transformation, transduction, conjugation and transfection is important. But we are human. When we understood that how bacteria or microbial world is actually functioning, we are actually using those techniques for our own needs, for our human needs. How? Here is the answer. We are actually doing such kind of things so that we can get the horizontal gene transfer. Okay, we can get horizontal gene transfer. For example, here is one DNA. Ultimately, what will happen? In one side there will be some replication from one DNA to another, in other side transcription will be happen from DNA to mRNA, and from mRNA to ultimately you will be getting after translation through ribosome you will be getting the ribosome that is a mRNA ribosome complex, and then you will be getting the after the protein folding and all such things you will be getting some biological active protein. For example, insulin, let me write clearly. For example, insulin, you need insulin and you know the DNA or particular gene which is producing insulin. What you will do? You will take particular part of that insulin causing gene and you are going to insert as a foreign DNA or RNA into bacteria or eukaryotic cells. How? By using these things, by using these things. We call it as a horizontal gene transfer, and when your gene or your insulin gene will be inserted among them through the plasmid, they will translate. When it will be translated, it will be create certain protein, and that protein will be insulin. We are doing such things in our 21st century in a very rapid manner. That is why microbial genetics become very much important part for our understanding. So, first of all, here I have mentioned a lots of that is a term transformation, transduction, conjugation. Okay. Let me tell you one by one how they are happening and what are the differences. So, transformation. So, transformation is a uptake of genetic material from the environment by bacterial cell. Uptake of genomic material, uptake of genomic material from the environment by bacterial cells. See, bacterial cells are actually living in this planet, and at the beginning I have told you they need to save themselves from a lots of that is a environmental factor. So, what they do? They actually try to uptake certain that is a element from them. So, for example, there are two ways we can do uptake, but before that let me tell you, in nature the genetic material often comes from the adjacent lysage bacteria and can include plasmid DNA or fragmented DNA released into the environment. So, there are two ways you can do this if you do in the lab, but in nature naturally in two process it is actually happening, either the lysed bacteria or through plasmid sharing. Okay. Naturally it is happening, but we are human, we know the science, we are actually using these things for the beneficial of the mankind. Okay. Basically for human, we are actually trying to use the transformation. So, how we can do? Either in chemical transformation or electroporation. This is a chemically modified that is a component cell, and here you have some other plasmid or something plasmid, some genetic information. So, you are going to give some heat shock, you are going to give some heat shock for at least 42 degree centigrade for 30 seconds. What will happen? Some pore will be created here, you can see some pore was created, then this thing actually inserted, and then after recovery you will be gaining one bacteria with this plasmid, you can see here. Another way electroporation, here is a electrocomponent cell, or we can say electrocomponent bacterial cell. We are doing some incubation for certain time, after that we are going to give electric shock at least 15 kilovolt per centimeter for at least 5 microseconds pulses. What will happen? Some pore will be created and automatically it will be entered. Your genetic plasmids or genetic component will be answered, and then you will be getting the recovery like this, whatever happening here. So, this two process, other process also here I have mentioned only two. So, this two process, the transformation actually happening. Transformation is just simple, transformation is very simple, transfer the genomic material into this. Now, the various factors that promote natural transformation in different bacterial growth, such as growth phase of the cells or presence of the specific substances. Though not all the bacteria are naturally competent to take the DNA, they can be made competent through chemical manipulation on lab. See, some bacteria they will be not very easily, for example, this genomic material, this plasmid, you want it should be within the bacteria. Naturally it is happening in various process, but all the bacteria will not follow the natural process. In that case you have to do chemical transformation, or you have to do the otherwise electroporation. So, this commonly done using by calcium chloride using permeabilized cell membrane. So, the bacteria can easily uptake our plasmid of interest. Scientists can also use electroporation, whatever I have mentioned, application of electroporation, charge to cell to increase the cell membrane permeability and thus transformation efficiency. So, this is something about the transformation. Now, in my previous slide, here I have also mentioned another word called transduction. You have seen what is transformation. Now, let me tell you about the transduction. What is transduction? So, it is a process by which a virus can transfer genetic material from one bacterium to another. Here one virus is actually playing a big role. Right now all of you are might be as we are recording this lecture in COVID pandemic, all of you are might be highly worried when you listen the word called virus. But my dear students, you have to understand all the viruses are not harmful. There are some good viruses are also present. Can you tell me any example? T2 bacteriophage virus. So, some good viruses also here, but maximum of them are not that is good for us, they are harmful. So, here viruses are actually playing a big role. Viruses like bacteriophage are able to infect bacterial cell and use them as host to make more viruses. After multiplying, they are actually these viruses are actually assemble and occasionally remove a portion of host DNA's host cells bacterial DNA. For example, you can see this is the bacteriophage, okay. And what happened? You can see this part, this violet color part actually viral DNA, and these things, this green part, this is actually bacterial DNA. So, what happened? When it has tried to penetrate and it has attacked to this bacteria, ultimately it has released its genomic material. You can see its genomic material to the bacterial cell. I hope you remember in my previous lecture I have told you that in bacteria the genomic material it is open within their cell, there is no any nucleus, these are not protected, this is open. Due to that, you can see here the bacterial DNA, these things bacterial DNA and viral DNA, they are actually coming closer, and due to that when it will take over or it will that is take over the entire cell machinery, virus DNA it will be get translated to create new viruses. Obviously, that time there is some transaction will happen. You can see here new viruses actually created. Let me remove all this ink. Here you can see here is one virus, it has produced itself in a multiple number, and then you can see here this virus, its genomic material is completely different than other viruses. Okay. So that this virus created similar virus, but also created another virus having some part of DNA from bacteria. When this virus having the bacterial genome as well as the viral genome, when it will infect another bacteria, what will happen? It will also gonna share some part of its DNA within this bacteria. So, this is how bacterial transduction is actually happening. Basically, virus is actually playing a big role. So, that is why after multiplying, these viruses assemble and occasionally remove portion of a host cell bacterial DNA. And later, when one of the bacteriophages infect a host cell, whatever I have mentioned, these pieces of bacterial DNA may incorporate with the genome of the new host. There are two types of such kind of transduction, one is the generalized, one is the specialized. Now, what is in generalized? In generalized transduction, the bacteriophages can pick up any portion of the host genome, and in specialized transduction, the bacteriophages pick up only specific portion of the host that is a DNA. Okay. Only specific portion, that is why you call it as a specialized, and here any portion, general case any portion. So, maximum case it was found by the scientist that generalized that is a transduction, it is taking much more part. So, scientists have taken advantage of the transduction process to that is a stabilize, introduce gene of interest to various host or that is cells. Transduction is a common tool by using by scientists introduced to different DNA sequence to interest of their interest into bacterial host genome. Today, how they are doing to do this? Scientists actually use this thing called phagemides, a DNA cloning vector that contains both bacteriophage and plasmid properties. These phagemides are packed into replication incompetent phage particle with assistance from helper and phase period to the transduction. Scientists also use transduction to introduce foreign DNA into eukaryote cell like mammalian cell line. These things are actually going. I can do, if I tell you more, this can be done by lentivirus or adenovirus or lentiviral or other kinds of viruses. Why? Because the lentivirus or adenovirus can be used where a gene is actually gene of interest is expressed, but not integrated into genome, and the stable cell line where where foreign DNA is actually incorporated. Okay. And all kinds of different lentivirus or that is adenovirus or adeno-associated plasmids are as well as ready to use viral preparation to add a new gene. So, this was something about the transduction. Okay. I hope it is very much clear to you all how the transduction is actually happening. Now, let me go to you another thing called conjugation. What is conjugation? It is very simple. It is a process by which one bacteria transfer genetic material to another through direct contact. You can see this is the bacteria, this is another bacteria, they are coming in contact, and through this contact it is actually transferring some parts of its plasmid, and you can see it has having this new information. So, during conjugation one bacteria serves as a donor, another serves as a recipient, and donor bacterium carries the DNA sequence called this thing called fertility factor, F factor. F factor allows the donor to produce a thin tube like structure called pilus, which actually which the donor uses the contact of the recipients. The pilus then draws two bacteria together. I am basically telling about this draws two bacteria together, which at a time donor bacterium transfer genetic material to the recipients. Typically, the genetic material is in the form of plasmid, a small or circular piece of DNA. I will tell you more in my next slide what is plasmid. The genetic material transferred during the conjugation often provides the recipient's bacterium. Okay. Here is the plasmid, here is the picture of the plasmid, you can see this is the entire bacterial picture, this is the plasmid. So, the plasmid is a small or circular double stranded DNA molecule that is actually distant from cell's chromosomal DNA. The plasmid naturally exists in the bacterial cells, and they also can occur some eukaryotes, and often genes are actually carried in plasmid to provide bacterium with the genetic advancement such as antibiotic resistance. Now, let me tell you more at first what is just a second. So, I will tell you about antibiotic resistance in my next slide. Before that, let me tell you more about the plasmids. This plasmids has a wide range of length from roughly 1000 DNA base pair to 100 of the 100,000 base pair. When a bacteria actually divides, all the plasmids contained within the cell are copied, such as daughter cell receivers from a copy of each plasmid. Bacteria can also transfer plasmids to one another through the process called conjugation. Okay, we have seen what is conjugation. But plasmids are sometimes also playing a very good role in recombinant DNA technology, where we have a gene and we have a plasmid, we are actually joining together this DNA, this gene and plasmid. Together we call it as a recombinant DNA. But I have told you about the antibiotic resistance. What is antibiotic resistance? This is very simple. Due to plasmid, it is actually happening when a high number of bacteria, a few of them actually resistant to antibiotic. What will happen in next step? Antibiotics kill the bacteria causing the illness, as well as good bacteria protecting the body from infection. See, many of the antibiotics are actually beta lactam based antibiotic. They are actually stopping the synthesis of the bacterial cell wall. Due to that, antibiotic cannot identify which one is good, which one is bad. Okay. Due to that, they always kill all the bacteria, and due to that some bacteria become the resistant, and then bacteria can even transfer the drug resistance that is a plasmid to another, and it is create a problem. How this is happening? You can see through either conjugation or either we can say transduction, or sometimes bacteriophage is actually also playing a big role in that is antibiotic resistance. So, it is a very big problem in present day scenario. In every 45 seconds a person dies due to antibiotic resistance. Now, if I tell you more what is antibiotic resistance, what are the antibiotics are present with us, these antibiotics will not work in case of antibiotic resistance. Okay. So, nearly 7 lakh deaths are actually happening in the all over the world due to antibiotic resistance, and it is predicted that in up to 2050 there will be at least 10 million people or 10 million deaths will be happen by that is due to this antibiotic resistance. What are the causes? Over prescribing of the antibiotic, patients not taking antibiotic as a prescribed, or unnecessary antibiotics actually used in the agriculture, or poor infection control in hospitals, or otherwise in many of the cases it is observed that poor hygienic or sanitation practices or lack of lab test. Due to that this problem is actually rising. Okay, we have to take care of this. How can we stop? We can improve our lab, we can collect share data, we can also use antibiotic wisely. We should not prescribe any of the antibiotic. Okay, we should stop such kind of practices, and then take necessary measures to prevent the infection. So, this is about the story of antibiotic resistance. Now, let me just cover the another thing is called transposons. What is transposons? It is a class of genetic element can jump to different location within a genome. Although these elements are actually frequently called as jumping genes, they are always maintained in a integral site of the genome. Transposons can found as a part of bacterium nucleate or in a plasmid. They are usually between 1 and 12 gene long. For example, you can say here is DNA A, this is DNA B. This part it can jump, it can insert here. Okay, you can see it is actually jumping, it is changing its position. That is why you call it as a jumping gene or transposons. Transposons contain a large number of genes including antibiotic resistance or other traits, flanked both ends by insertion sequence coding for a enzyme called transposase. Transposase is an enzyme that catalyzes the cutting and resealing of the DNA. Whatever is actually cutting is happened, this was actually done by transposase. Okay. Thus, such transposons are actually able to cut themselves out of the bacterial nucleate or plasmid and insert themselves into another nucleate or plasmid and contribute in the transmission of antibiotic resistance. Okay. So, today we have learned all those things. We have seen what is microbial genetics, we also covered transformation, we also have seen transduction, conjugation, plasmid. Along plasmid we also have seen AMR, antimicrobial resistance. Then I have ended up the lecture with the transposons. So, that is it from my side. In next lecture tomorrow, we are going to learn about the microbial biotransformation. There are lots of activities, lots of stories of biotransformation is actually happening. So, what is microbial biotransformation? How microbes are helping in transforming something biological. I am going to tell you about it in detail in my next lecture. We are going to see what are the types, what are the advantages, what are the disadvantages application of microbial biotransformation. So, all these things we are going to cover in our next lecture. So, thank you all of you for joining this lecture. I hope all of you have enjoyed my whole lecture and you are learning very seriously, and all the things whatever you are learning, I hope it will be helpful for you in your upcoming examination, and also if you become a researcher, all the things are going to help you in your research activities. So, thank you all of you for joining. I wish the best to all of you. God bless you all. See you in my next lecture. Thank you.